All right, hi everybody, and welcome to the first lecture of Biochemistry 110, which is Biochemistry and Society. So this first lecture is just going to be titled Biochemistry and Society. So it's pretty much an introductory lecture to what the kind of things that we're going to be talking about throughout the entire course. So we'll start out the lecture just talking about society. What is society? Some sociological terms, some history of human societies, just so we're all on the same page. Then we'll move on to what is biochemistry. So what is biology? How does biochemistry fit into that? And what are some applications of biochemistry? So applications in medicine, nutrition, agriculture. And then we'll go on to do basically what we're going to be doing for the rest of the course, which is talking about different issues in society and how biochemistry intersects with these. So the point of this course is not to memorize things or pass any tests. There's no tests that you really have to pass. It's nothing you need to memorize. This is all very low stakes. You just need to sit back and try and learn some things and maybe think about some things that you haven't learned about before. So the nice part about this class is if you can have an understanding of biochemistry, it can really change how you see the world around you just in your day-to-day, everyday life. So those are the goals for the course. So let's go ahead and get started with the first lecture, Biochemistry and Society. I hope you all enjoy it. So this course is called Biochemistry and Society. So we'll start out the course by going over some general terms just so we're all on the same page in terms of what we're talking about when we're talking about society. So I, of course, am a biochemist. I'm not a sociologist, but I do have an interest in the topic. And so these are just general terms. Some of them you may understand more than I do, but I think it's really important to define them right away. So first of all, what is society? So a society is a group of individuals who share some degree of social interaction and are connected by likely some sort of geographic and cultural norms. So we have these images right here. So a family would be considered part of a society. Obviously, these are people you could have very close social interactions with. You may live in the same house. There may be multiple generations, and this may differ between different societies and between different families within the same society. You could also picture something like a village right here, which would be a smaller society, a smaller group of people interacting. Maybe they sort of know each other's business, have maybe a rivalry with similar villages that, have generally the same cultural norms, but maybe they differ a little bit from village to village. You could also move up to larger cities where lots of people may interact, sharing the same space and stuff like that, but may only have sort of surface interactions with each other. You may have your own society within that city, your own cultural group, your own everything like that. But that would be a larger part of society, and you can go even further to that. So here we have a picture from space of the Nile Delta. So this would be a larger region. And there's been societies in this region sharing different cultural norms throughout history. And this, in the modern time with modern communications, can be even expanded into a global society where we all do interact in some ways, even within our, from the context of our respective cultures. So societies are groups of individuals who share some degree of social interaction and are connected likely by geography and cultural norms. So the social behavior that arises in humans that, gives rise to societies can also be observed in other non-human animals. So social behavior or social ethology, which is the scientific term for the study of behavior, is quite common in a lot of different animals, and this varies from different species to species. So a lot of different non-human animal species do have varying levels of social behavior. And so this is where individuals will behave with a regard for the group at a large, or for somewhat related or closely related individuals. And so this behavior, this consideration of the larger population or the larger group, and the modification of your individual behavior based on that can actually increase the individual's fitness and survival, which is what all of sort of evolution or evolutionary genetics is sort of based on. So ants are a very good example of this. So ants actually function, basically the organism of an ant is basically the ant colony. So some ants, I believe, aren't even most of the individuals aren't even, sexually capable or of sexual reproduction. And so they're just a part of what is the larger organism. So they have communication with each other. There's like trophallaxis, where they sort of, will spew out chemicals to sort of have communications with each other. And so their lives are very well coordinated with this social behavior. Bees are very similar, so they have to coordinate for their survival of the hive. So they have to work. It would basically, one bee cannot survive. A bee can only survive in the context of an ant colony. So they also have behaviors such as communication dances and stuff like that, and shared responsibility of raising larvae and making food and stuff like that. So these two species, which are in the Hymenoptera family or order, uh, show a great deal of very interesting social behavior. But even larger animals, such as non-human mammals that aren't really related to us, also show a lot of social behavior. Down here, we have a picture of a pack of wolves try at least attempting to take down a larger bison. So this is something that a single wolf could not do on its own. It would have to coordinate its behavior with other wolves in the pack. And so this behavior uh, ensures the survival of the individual more than if it was just a lone wolf, as it were. Killer whales, another really interesting example. You can see here's this really interesting picture of at least four killer whales coordinating their activities together to, looks like, knock this poor seal off this iceberg so they can either eat it or train each other to do this type of behavior in the future. It looks like there might be a little baby killer whale right here. So maybe these older killer whales are showing this baby killer whale how to do that. This requires an intense amount of communication between these species to coordinate their movements like this. And it also demonstrates likely some sort of kinship or some sort of, these animals would have to have, care whether, you have to be able to work in a group. And so they're likely related. So they care whether they're closely related, siblings or mother or father or baby survive because their genes are related, and the survival of the group is sort of also passing on their own genes. So social behavior, which gives rise to human societies, is also very well and alive in other mammals, other animals, and even down to insects. Now, the best species to look at if we're thinking about human societies and human social behavior is, of course, to look at the great apes. So, humans are in the great ape family. These are our closest evolutionary relatives. So, the great apes all demonstrate varying levels of complex social interaction. So, the other great apes are orangutans, gorillas, chimpanzees, and bonobos. So orangutans are the least social of the great apes. So they're described basically as solitary but social. So they usually exist as solitary, as a solitary individuals who just sort of do their own thing, searching for food. So mothers will care for their babies, of course, for extended periods of time and have close social bonds. But they really don't. They really exist as solitary species for the most part. Although they do have overlapping territories for the most part and have complex communications that they're able to use. But for instance, there really isn't any like mutual grooming behavior that you observe in orangutans. Gorillas are probably the next most social, but less so than chimpanzees or bonobos. So gorillas do live in larger groups, and these will be controlled basically by a single male silverback. Although these, there's caveats to all of this sort of stuff. There may be lower ranking males in this group as well, but the silverback will basically have a harem of all the females that he mates with, and then the children that come from those matings, of course. The males can have weak social bonds, whereas those females within those groups could have stronger social bonds and have a highly competitive behavior as well. So gorillas do also show communications in their social groups, and they do have more sophisticated like play behaviors than orangutans, and they do have social grooming, which helps establish those bonds in these social groups. Now, humans' closest relatives are the chimpanzees and bonobos. We are extremely closely related genetically and last common ancestor to chimps and bonobos. So, if you go to the zoo and you see a chimpanzee, you're more closely related to that chimpanzee than an African elephant is to an Indian elephant, for example. So chimpanzees and bonobos live in highly sophisticated social groups that can be quite large and extremely complex. And this is obviously an active area of research for many, many years because it illuminates sort of how, for the same reason we're talking about it, it gives us insights into human societies. So chimpanzees are known for their sort of more aggressive behaviors. So chimps live in groups of 20 to 150 members, which is a very interesting number, I think, for reasons we'll get to in a minute. But they will break out into smaller temporary groups. So they have to be able to establish these social ties and exist as groups up to 150 members. These groups will have a lot of different ages and sexes of chimpanzees. So it's male dominated, definitely, but there's not just basically one male and a bunch of females. There's lots of different males, which makes it a lot more complex. They also, unlike gorillas, which basically just sit around munching on vegetation all day, chimpanzees have to be able to coordinate and hunt for food. And so these are male dominated groups, and there's a linear dominance hierarchy. So, one male will be dominant over another male, will be dominant over another male, and so on and so forth. And within these groups, you can form complex alliances. So, like coalitions. So, just think of, alliances like on Survivor or something like that. But it is extremely complex. So even if you're a lower ranking male, that can actually be beneficial for sexual, opportunities and stuff like that, because no one's really paying attention to you. You can kind of sneak in there. But chimpanzees, one thing that is characteristics of them is that they are highly territorial, and they will kill chimpanzees from other groups, and they will regularly display engage in infanticide as well, which has to do with making sure that it's your genes and not some other chimp's genes going off into the other gene pool. So chimps are known to be a bit more aggressive, highly territorial, and that's partially thought because they live in regions where food is a bit more scarce and they have to maintain these large groups. Now, bonobos are interesting. So they used to be called pygmy chimpanzees, even though they're about the same size as other chimpanzees. But they are known for being way, way less aggressive than chimpanzees. And bonobos also have, interestingly, female dominated social structures. So there's societies or groups that they live in are about the same size as chimpanzees, but they're much less territorial and much less aggressive. They're also known for being highly sexual. So they're sort of it's been said once that chimpanzees use violence to get sex, whereas bonobos will use sex to avoid violence, which is a very interesting distinction between these two groups. So bonobos tend to live in places that are a bit more food rich or resource rich. So they have much less defined territories. They're not going to just kill other bonobos because one of them, wanders off into the other one's territory. So food is more easily found. And this female dominance of the society is also very interesting, to the point where females are actually able to sort of bully males into hesitant males into having sex into mating with them. Males don't really perform infanticide as well in bonobo societies. They're very tolerant of juveniles and infants. And mother son bonds are very, very strong in bonobo society. So what else can I say? The sexual behavior then is very interesting. So it's thought that bonobos and humans are the only species that are known to have sex face to face, which is quite interesting. And they also do the thing where that bonobos or chimpanzees do, where they live in these larger groups, but they'll split up during the day and then sort of come back in the night. So we can go on and on about great ape social societies, but it is important to touch on them and define it a bit because they are our closest relatives. So if we're talking about human societies, should mention our closest relatives, and you see that they have a lot of differences between even closely related species such as chimpanzees and bonobos. But all of this behavior is very reminiscent of human societies, and why studying the social behavior of our great ape relatives is such an active field of research. Now, something that I really want to emphasize, and I think is a really important point for just understanding human societies in general, is that humans we have lived as hunter gatherer societies for most of our history. So, anatomically, modern humans, the dates range, but let's say somewhere between 100,000 and 150,000 years ago is when you could say, oh, this is an anatomically and somewhat behaviorally modern human. If you took a human person from 100,000 years ago, plopped him, gave him a haircut and some clothes and a bath, and plopped him down in the middle of Times Square, you really wouldn't. They would just blend into the crowd. And so most of human history took place in hunter gatherer societies where you would basically live in groups. We're guessing between something like that chimpanzee number of 20 to 150 200 individuals. That seems to be the consensus number. And you'd basically gain sustenance from hunting wild animals and collecting and eating wild plants. So nothing was domesticated. There was no farming in terms of either livestock or plants as well. So there's a few things that we do know about these types of hunter-gatherer societies, or assumptions that can be made based on evidence. Of course, they're nomadic or semi-nomadic. So unlike later in our history or now, you might grow up and live your whole life in sort of the same town or city or something like that, or same general region. Hunter-gatherer societies were not like that. You had to follow the sort of food that is available. Another important part is that these societies were generally very egalitarian, or much more egalitarian than the societies of humans that came later in our history. They'd sometimes be described as fiercely egalitarian, where if you did not share things as well as other people, you could be kicked out of the tribe, which would be a terrible, terrible thing. Humans basically can't live uh, outside of society in these social structures. So if you think about today, what's basically the worst punishment you can give to the worst criminal is to lock them up into solitary confinement. This has been described as cruel and unusual punishment because people basically go crazy and lose their mind if you're locked up in solitary confinement. These people may be murderers. They may want to murder people when they're interacting with them, but everybody wants to interact in a social group, to some extent. And this likely, of course, goes back to our history of existing as these in a hunter-gatherer society. So unfortunately, there's a lot we don't know about how humans used to live in these hunter-gatherer societies. And the information that we do know comes from sort of remnants of hunter-gatherer societies that remain around the world. So here we have some of the pygmy people of Central Africa in the Congo region. There's the San people of South Africa. There's also people who live more hunter-gatherer societies in Papua New Guinea, and of course in the Amazon rainforest in sort of these remote regions. So the information that we have about our distant past in living in these hunter-gatherer societies, however, it is extrapolated from studies on how these groups sort of live. We don't know that, and the one thing about these regions is that they are often quite resource poor, or like protein poor or something like that. Um, and so we really don't know how people may have lived as in hunter-gatherer societies in a more fertile land, let's say like the Indus Valley or something like that. Um, One other thing, so I talked about the fiercely egalitarian system. So that one of the ways this is outlined is in this picture here. So obviously, the men in this small village went on a hunt, and then they're splitting up and dividing the meat very equally. So that this fiercely egalitarian system is something that's very reminiscent. That's very characteristic of hunter-gatherer societies. And so another example of this is shown right here. So this is a picture of what's called a potlatch from the Aboriginal people of the Northwest United States, or the Northwest Pacific region in the United States and Canada. I think this is the Tlingit people, if I'm pronouncing that correctly. So these societies were actually very resource rich. So you had these very dependable or predictable salmon runs that you have every year. You just set up a bunch of traps, gather a bunch of salmon, dry it, and you're good to go. So these, although they are hunter-gatherer societies, they could you could generate quite a bit of wealth in these societies. And when one group or one clan or one family generated too much wealth, they would have what's called a potlatch, where you basically invite everybody over, have a feast, and give away everything that you can. And it's a way to keep these types of societies more egalitarian. So it's easy, and especially during the early years of anthropology, it was there was the people doing that type of research looked down on groups like this, but really you shouldn't do that. There's a lot of evidence that they could be, that this way of life and these people could be superior in a lot of different ways. Number one is in intelligence. So uh, Jared Diamond said that basically the smartest people he ever met were people living in somewhat hunter-gatherer societies in Papua New Guinea. So if you think about your life, if you need to go get food, you just, drive over to the store. It's all done there. It's all done for you. I don't even know how to fix my car if it breaks down on the way to the store. But if you live in a hunter-gatherer society with people, you have to have an intimate and extensive knowledge of your environment. So what foods and mushrooms are safe to eat, when they're not safe to eat, when they appear at what time in the year, the behavior of animals that you're hunting and stuff like this. This requires an incredible amount of intelligence in these societies. And also you think, oh, well, that must have been a ton of work. Well, specifically when you're talking about like the San people in Africa, basically they can have all their needs met, but by doing just like 5-6 hours worth of work a day. So I think a lot of us would be jealous of that. So hunter-gatherer societies, also a very, very interesting topic. I'm not an expert, but it is an interest of mine. And this is the type of society that humans lived in for most of our history. So when did this all start to change? When did humans shift away from hunter-gatherer societies? So that happened during what's called the agricultural revolution, or sometimes called the Neolithic resolution and revolution. And this occurred when humans began over a long period of time to start to domesticate and farm plants and animals. So this started about 12,000 years ago, is the basically the oldest accepted date. That corresponds with when the end of the last ice age, basically when the glaciers that covered most of the world, at least the northern hemisphere, began to retreat and open up a lot of different new lands. This spurred an agricultural revolution independently in lots of different areas across the globe. So the dates for this vary significantly. It was a lot earlier in some regions and a lot later in other regions, but over time, sometimes probably over thousands of years, humans started to domesticate different plants and animals. So this slowly spurred a shift from hunter-gatherer societies to larger, more complex and geographically stable, so less nomadic centers. So the first documented place where plants and animals started to be domesticated is in what's called the Fertile Crescent of the Middle East. So, what this is disregarding dogs too. So, dogs were domesticated probably way before the agricultural revolution. So, they really are man's best friend. But sheep and goats specifically in this region were the first livestock to start to be domesticated, and you can sort of picture how this would happen. Maybe you would, some industrious person would think, well, here's this herd of sheep that I keep following. Maybe I can build a pen or something to keep them all in this area. Then, oh, well, there's this really aggressive sheep that's causing problems in this flock. Is it a flock or herd? Let's get rid of that one and then keep these more peaceful, docile ones together. So over years and years and years, generations likely, of course, this resulted in domestication first of sheep and goats in this Fertile Crescent region. The first plants that were domesticated in this region were likely wheat. So these are quite easy to grow. They're just grasses. And so you can also sort of use your imagination and imagine how that would start to happen. Oh, there's these really nice grasses that they have these really thick, starch rich, endosperms on the end of these grasses. Let's take some of these seeds, throw them in the ground. Remember this place. Come back the next year, and then eventually you'd take the biggest seeds with like the best stems that's capable of holding those seeds, and pick those. So it likely happened a combination of sort of by accident and with purpose. So barley and wheats type things were one of the first ones domesticated in this Fertile Crescent. So domestication also happened at lots of other different areas. We have the Southeast Asian region here. So pigs were domesticated in this region, as well as chickens, ducks, chickens, and also rice and things like taro and stuff like that were all domesticated in that region. China was another region of domestication. So barley, buckwheat, different plants like that in that region. There's several regions in Africa. So in this region right here, barley or coffee, cotton, sorghum, those are really important ones. This region here in West Africa. I don't know if this is the best map in the world, but it's the nicest one that I found. Stuff like millet and oil palm. And then also the Americas was a huge place for domestication of specifically plants. There wasn't a lot of animals domesticated in the Americas except for guinea pigs, llamas, some duck, and maybe the turkey, and definitely a dog. But plants that came from regions in Mesoamerica, so in Mexico and Central America, and regions of North America, and especially the Andean highlands and apparently in eastern Brazil. The number of plants that you recognize from this region is huge. So we have blueberries, cranberries, sunflowers. We're in the sunflower state. Tobacco, beans, corn, chili peppers, tomatoes, squashes, pumpkins, and potatoes. That's a big one. That was in the Andean region. So that's why the Columbian exchange, after Columbus discovered the New World, that's when Italy and China and India basically gets, tomatoes for pasta sauce. China gets, spicy peppers, and India gets spicy peppers for all their types of food as well. So agricultural revolution, this happened in many different times over a long period of time. There's a lot to think about in this topic, but it's basically the important part for this is when society started to shift from hunter-gatherer society to more stable, geographically stable, larger societies that sort of we exist in to this day. So you could spend a whole course talking about the benefits and drawbacks, or the perceived benefits and drawbacks of living a hunter-gatherer society to a more agricultural pastoral society. Um, but what is known is basically humanity, or human societies did change significantly after the agricultural revolution. So the conversion from hunting gathering to agriculture had significant consequences on societies. So one of these is population size. So obviously, if you're able to make a large amount of food that's somewhat reliable, and you could store that for the winter or for the next year, that definitely increases the amount of humans who can live in a particular region. So during in the Fertile Crescent, during the beginning of the agricultural revolution, that's the first time in human history where you basically have this shift from humans living in, let's say 20 to 300 people. Now you can have, this is the first time where you see cities where you can have actually thousands and thousands of people. So this is actually from the Indus Valley civilization right here. And they're able to work together because they have enough food reliably, and because they have a good reason to stay in the same geographic region and probably want to protect that region as well. Another result of the agricultural revolution is that you see definitely an increase in, I'm saying general knowledge here, but more just technological advances. So again, with the Indus Valley system right here, they had a complex, this is basically a swimming pool right here. So you had time and resources to think of, basically make more complex sort of things to increase the quality of life of individuals right here. This is also when complex writing started, or just writing systems in general started to occur. Of course, oral communication and oral histories have always are an integral part to just the human animal in general, but writing systems actually arise in this time. You can also get more complex artwork. So we find, cave drawings and small figurines and stuff from 20, 30, 60,000 years ago. But this time now you're actually able to support an like an artisanal class that's able to make these complex sort of artworks. However, is probably the change from hunter gatherer to agricultural society was not always a positive thing. So social inequality definitely changed significantly over after the agricultural revolution. So this is now when you get class. So you're no longer in a society that's fiercely egalitarian. In fact, you're living in a society where hierarchy is a very important part of it. So if you have this land and this house and this city, you want to make sure that is yours. You have enough labor below you, and they have an incentive to work that land, because you don't want to work it, and you also want to pass that land off onto your offspring. So gender inequality also seems to take a big hit for women in these types of societies. Especially since now you definitely want to pass your possessions and your land off to your offsprings. You better make sure that you sort of have control over your wife, or the women who you're breeding with, to make sure that her children are basically your children. It's a very complex topic, and this is when we first sort of see slavery enter in society very early on, and of course that lingered for way too many years, of course. Um, the agricultural revolution also did make people more vulnerable to natural forces and diseases. So if you're living in a specific area and you're depending on that crop being good next year, but there's a flood or a drought, that's not good. If you live in a hunter gatherer society, that's just part of life. You know that there's a flood in this region and there's a drought in this region. You can move to a different region to find the foodstuffs that you need. In permanent agricultural societies, that's no longer an option, and you see that climatic changes often will definitely led to the complete dissolution of certain societies. Also, diseases at this time. So, most, and we'll talk about this in a later lecture, but most diseases come from animals that have been domesticated. Almost all the diseases that we deal with on a day-to-day basis or over generations come from, they basically jumped species from domestic animals. And also nutritional decline. So if you imagine you're living in a hunter-gatherer society, you want to go get some food. Well, maybe you eat it, you check your traps, you have a squirrel, you check your go to the stream, you can have a small fish that you had in there, and you know that there's wild mushrooms, and there's some, wild mushrooms and some wild onions and some wild potatoes that you can eat. That's a pretty complex nutritional diet. Slaves in Egypt were basically just drinking beer and mushy bread and stuff like that. And so that's when you start to see even the pharaohs in Egyptian tombs all had terrible tooth decay and diseases and stuff like that, and nutritional deficiencies. So agricultural revolution definitely changed society in a lot of ways for the better, or just, it's just what happened. There was no going back once you started these societies, but also has some negative consequences that should be definitely considered. And the next big change that happened in human society basically occurred from the mid 1500s and is still, I would argue, going on today. And so these would be the scientific, industrial, technological revolutions that occurred over the past few hundred years. So the scientific revolution, which started with Copernicus and Newton, that is generally dated to the mid 1500s. This is when humans first developed the scientific method as a sort of discipline, and that definitely changed and spurred on what was called the Industrial Revolution. So the first Industrial Revolution started around 1760 and went to 1830. And then there was another technological revolution from the 1870 to the 1910s. So this gradually shifted humans away from agrarian society into these manufacturing societies that we sort of know and live in today. So this at first occurred in England with the rise of, steam power and machine technology and factories and stuff like that. And this had a significant effect on human society, obviously. So one of the main things is this did have over time a significant increase in the standard of living of humans as a whole. So if we look over here to the life expectancy in 1800, basically most people the average life expectancy was in the late 20s to 30s. That's not great. So that does probably take into account the high level of child mortality, which is a part of human history for most of our history. But obviously in 1800s, life expectancies were quite low, and they've been rising ever since. So the life expectancy in 1950 is quite high, and now in 2015, humans are living longer than ever based on just the last 100 years of these technological interactions. However, there is a lot of negative consequences to the human societies making this shift. If you think about the early history of the Industrial Revolution, child labor was very bad. That social stratification in society, even though sort of all the boats were being lifted, working in a factory. I don't know if I've done it. It can be pretty miserable, tedious work, although farm work can as well. And then things like, war machines and stuff like that. Now with the significant scientific and manufacturing advances, war became much more consequential and much more you're able to basically inflict a lot more pain onto a group of people. So in hunter-gatherer societies, there's a limited amount of pain that one group can inflict on another. Earlier society, and this is all the caveats to it, earlier societies as well. I mean, with knives and shields and arrows, there's only so much you can do. But when you're talking about bombers and atomic weaponry, that's a big difference as well. So I'm even less of an expert on this period of history. But obviously, human society changed significantly with these types of advances. And after the Industrial Revolution, basically what we're going through now in a lot of different societies is what would be called a digital revolution and entering into what is a post industrial society. So after World War II, especially in the United States, if we just keep it at that, there's a shift towards digital technology rather than machine or analog technology. And that has ushered us into what is called the information age, where a lot of us, myself included, basically make our livelihood not off of extracting materials or even manufacturing materials from those raw materials that are extracted, but with ideas or service based sort of technologies. So this is Clark's sector model that sort of shows over the years the decline from the primary sector, which is basically the raw material extraction. So that's, mining, fishing, farming, logging, stuff like that. And even a decline in so that would be the decline in primary sector. And then, so this would be the decline in secondary sectors. So that would be manufacturing. So that would be taking fish and canning it, or taking timber and turning it into paper. And then with the tertiary or quaternary sectors, which are, sort of information, more service, which I believe is tertiary, and information based, which might be quaternary sectors, increasing over that time. So that's where basically the society that we live in now, where most of the society is in a service or information based technology rather than extracting raw materials and using them to manufacture products directly. So this is another important feature to understand where we came from and where we're going in terms of human society. Now, another really important part of society is that we've already touched on a little bit, is sex and gender or gender roles in society. Now, obviously, this is an evolving philosophy these days. So if I say anything incorrect, yeah, I'm happy to. Always happy to learn about things and ideas that I'm not well versed on. But it's sex and gender roles are definitely very important in society and have evolved in different societies over time. So, sex or gender roles can basically be described in the simplest terms as behaviors which are appropriate for a specific sex or gender in a specific society. So, obviously, since the beginning, even in hunter-gatherer societies, the fact that females can bear children obviously alters their roles significantly from males in that society. But we can see it's a very interesting topic in a lot of different stuff. So we got this guy right here. I believe it's Louis the 14th, the former king of France. And you can see how he's dressed here very extravagantly. He's got these tights down here and these awesome shoes right here, which would be considered quite feminine these days. But really, back in Louis XIV's time, he was considered the absolute height of masculinity. He was considered an extremely virile and masculine person. So you can see, obviously, that these sorts of constructs are evolve and modified in different societies at different times. Really good example of this, I was watching the show A League of Their Own last night, which is a really interesting examination of the changing of sex and gender roles in societies around World War II. So women entered the workforce in numbers that hadn't happened in a long time in the United States during World War II, with the men off to war. And so women were able to fill roles in societies that needed to be filled just out of necessity. A similar situation happened after the genocide in Rwanda in 1994. A lot of the men in that society perished, and so women actually filled a lot of government roles, and they do to this day. So it's a role in society they weren't able to fulfill prior to that, just out of necessity. And it's also very interesting. There's a lot of societies, including Samoa, which accommodates for a third gender. There's a specific term for it that I don't want to butcher, but these are individuals who have basically identify with both characteristics of masculinity and femininity. a similar situation occurred during in the Plains Indians during historical times, where if men just straight up were not into hunting or riding horses or anything, it was perfectly acceptable for them to have a role in society and do basically work that was considered more appropriate or more usual for females in that society. And we will definitely talk about this lecture in terms of biochemistry in society, because the role of sex and gender roles in society can be influenced by biotechnological advances, including birth control, which we'll talk about a little bit later. So kinship and ethnicity is also a really important part of maintaining different societies' identities. So all human societies recognize different levels of kinship. So this includes parents and children, marriage, so that would be considered kinship, and even adoption. Basically, every society has allowances for adoption, and you're considered kins. So a lot of larger societies are organized or based on ethnicity. And so ethnicity is a social category that basically distinguishes people based on things like shared ancestry, religion, language, culture, etc. So some nations are more multicultural, like the United States or England. And some nations are pretty homogenous ethnically. So Japan is something like 98% of citizens of Japan are actually Japanese. So the largest ethnic groups on the planet, number one is the Han Chinese group. Number two is our Arabs. It's number two, and then Bengalis would be number three. And so this is just an important topic in when you're talking about society, but also biotechnology comes in, or biochemistry comes into this as well. So if you remember a few years back, there was this movie Eurovision starring Will Ferrell and Rachel McAdams, and it took place in Iceland, even though neither of them are Icelandic. But the running joke throughout the whole movie was that they were, they loved each other or were interested, but they also were afraid that they might be related. That's actually is a problem in Iceland, and people are because it's just such a small population that came over to Iceland, and the country only has like 300,000 people. So if you go on dates, modern advances in biotechnology, such as genetic sequencing, you can do that before you really settle down and get to know someone, just to make sure that they don't happen to be a first cousin or something like that. So kinship and ethnicity, very important topic for society, and can be influenced by things like biochemistry. So government and politics is obviously also a really important part of when you're talking about society. Different societies are influenced by what types of government and political systems that they have. So basically the entire globe, with a few exceptions, has been carved up, and all the land in all in the whole world is basically under the jurisdiction of a specific government entity or entities. So this map here shows the differences in the level of democracy, basically, in each of these societies. Some are quite high and some are quite low. But government and politics can also intersect with biochemistry and society. So we'll talk about DDT later, which is an insecticide. And the United States actually had a ban on DDT through the action of its citizens appealing directly to the government on behalf of the environment. So this has worked better in some, the ability for individuals to influence government policy based on science. That is easier to do in a highly democratic society rather than a less democratic society. So during this course, please keep in mind, when we're talking about different nations, that the different governments and politics of nations differ between countries. And so trade and economics is also a huge part of society. So people live in individual societies, but since our earliest history, we've been interacting with other different societies largely through trade and economics. So it's grown more complex, of course, with the rise of large nations and very sophisticated communications. You can trade on the stock market across continents in matters, in milliseconds over the computer. But this is a very important topic in sociology as well. And so it's no coincidence that the Black Death plague, the bubonic plague, traveled from China to and basically ravaged Europe through established trade routes that had been around for generations and generations via the Silk Road. And so the bubonic plague was a bacteria, Yersinia pestis, shown here, which was transmitted by the rat flea, which is carried by rats. And that could only travel from this region in China over to Europe because there was these established trade routes. So it's another instance where these topics in society, you can see they are influenced by biochemical sort of ideas and topics. And we mentioned earlier how the Industrial Revolution changed conflict and war significantly. After the Industrial Revolution, basically, you went from, fighting with spears and arrows to swords and shields. And then now we have large, war machines that would be impossible without the advances of that shift in society from an agrarian society into an industrial society. And so the conflict and war is a very big part of all of our lives and the history of each of the nations in the planet. And biochemistry is actually, in the 20th century, biochemistry and advances in biochemistry had a huge significance on conflict and war. So really good examples of this is in World War I. That was basically the first fully industrial war, and mustard gas is a chemical that basically irreversibly affects your lungs and different visceral organs. And so this is a sort of famous portrait of soldiers in World War I who have just suffered a mustard gas attack and were left sort of blinded. Very tragically in World War II, the Holocaust occurred partly with the aid of Zyklon B, which was a chemical that was developed as an insecticide but was used on human beings in this war. And then the third picture we have here is of Colin Powell after the anthrax attacks in, I believe, 2002 or 2003. And so he's holding up a vial of anthrax here, which is a naturally occurring bacteria, but can be weaponized basically as a biological threat. So the threat of biological weapons is a huge topic nowadays. So biochemistry can definitely influence, and we'll talk about this more as the course goes on different conflicts and wars. So that brings us to the end of our discussion on sort of society and just some broad sociological discussions, just so we're all on the same page on the history of human society and different topics related to human society. So now we can start into the second part of this lecture, which is what is biochemistry? So the course is called Biochemistry and Society. We talked about what is society. So what is biochemistry? So biochemistry is the study of chemical processes involved in life. So we'll go into more detail here in a minute on this, but biochemistry is basically the most fundamental of the biological sciences. So if you think of biology, you might think of organisms, ecosystems, and their interactions. So organisms and populations of organisms interacting with the ecosystem, so with the natural world. So you have some bikers here, you have a plant, you got a pufferfish, you got some trees. So that might be that is one of the more broad aspects of biology. But you can actually go even a little deeper and think about cells, organs, and their interactions. So how does an individual multicellular or single cellular organism function? Well, it functions by the coordination of these different cells, which form organs, which form physiological systems, which allow you to carry out the different processes that you need in your everyday life. So biochemistry goes even deeper than that. So it looks at atoms, molecules, and their interactions that are involved in life. So what chemical interactions, so just inert chemical interactions, these cells are considered alive, but these chemicals are not considered alive, but they are the chemicals that maintain and sustain the processes involved in life. So we have water here. So all biochemistry takes place in water, so it's very important. We have a DNA molecule right here. We have a fat molecule right here, which forms the cell membrane around cells. We have an ATP molecule right here, which is the universal energy currency. So if you are running down the street, you're basically just burning ATP to provide energy. We have a tetrodotoxin here, which is actually a poison from a jellyfish. We have glycogen here, which is how your body stores excess glucose when you need it in different times. We have a lipid bilayer here that forms a membrane, and we have a protein molecule, hemoglobin, right here, which carries oxygen in your cells. So biochemistry is really fundamental to understanding how life works. So if you understand how these individual molecules work, you can understand how different cells and organ systems work, and that will help you understand how a whole organism or a whole population of organisms or a whole ecosystem of organisms actually functions. So we'll spend the rest of the time going into more detail on what is biochemistry. So we'll start with the most broad view of life on Earth. So of course, when we say life on Earth, that's basically we're just talking about life itself, because the Earth is the only place in the universe that humans are sure that life exists. Does it exist elsewhere? Probably, but we have no way of knowing. For all we know, life only exists on this one tiny planet next to an insignificant star in the corner of just another galaxy, as far as we know. So the biosphere describes the entirety of life on Earth. So it's the sum of all the ecosystems on Earth. So if we talk about the biosphere, we're talking about all known life on planet Earth. So the biosphere is a closed system in terms of matter. So with the exception of comets or meteors and stuff like that, hopefully not comets, but meteorites. It's a closed system, so there's not really any matter coming in or out of Earth. So life is a self-sustaining system. However, it is an open system in terms of energy. So the biosphere requires sunlight, which is used for photosynthesis, and that forms the basis of all life. So we have this really cool GIF right here, which shows the level of photosynthesis on each of the different geographically across the Earth over a year, I believe. So that includes both plant material. You can see here's the Andes right here, and the Central African jungles and the Southeast Asian and Indonesian jungles right here, which are very huge hotspots for vegetative matter. And then you can see the algae in the oceans, which is also critically important, and how these shift over time but remain relatively stable. So if we refer to the biosphere, which I will several times, this just refers to all living matter in all ecosystems on Earth. So what exactly is an ecosystem? So an ecosystem is basically the sum of all living organisms in a specific location, and it accounts for the abiotic environment with which they interact. So you have, a coral reef right here. They're interacting. This is the summation of all the living organisms. So all the fish, all the sea anemones, all the starfish, and all the microscopic organisms as well in this region, as well as the abiotic environment. So the salts and the water in which they interact, the rocks and the nutrients, the non-living matter. So abiotic means non-living matter. Here is, again, a forest, same sort of thing. You have the trees and a person walking around. So the ecosystem would be all the organisms in this forest or in this forest region and the abiotic environment that they interact. So energy always flows into the ecosystems by sunlight via photosynthetic organisms. And then ecology accounts and studies the nutrients that are cycled through these environments through photosynthetic and chemotrophic organisms. That is, organisms that don't do photosynthesis but live off eating photosynthetic organisms or other organisms that eat photosynthetic organisms. So that would be ecosystems. It's one of the larger views to look at biology. And we're not going to look deeply into evolutionary biology in this course, but biology only makes sense in the light of evolution. So the theory of evolution, of course, was developed independently by Charles Darwin and Alfred Russel Wallace in the mid-1800s. And since in the subsequent 150 years, evidence from so many different disciplines, so from genetics and biochemistry and behavioral biology and paleontology and geology, every single discipline supports the idea that all life descends from a common ancestor that existed, at our best guess, about 3.5 billion years ago. So if you have an understanding of evolutionary biology, it's really integral to understanding biology as a whole. So just wanted to mention it. So here would be the common ancestor, and then it broke off into basically the three main classes of life. So which are bacteria, which are single-celled organisms that you're all very familiar with, but there's a lot of diversity, long evolutionary history diversity within bacteria. The archaea, which are the second one, these are also single-celled organisms that are just as different from bacteria as eukaryotes, but they live in extreme sort of environments. So I think they were first discovered in hot springs in Yellowstone. And then there's the eukaryotes or eukaryota. And so this is the family in which we belong. So these can be single-celled organisms. So like yeast are single-celled eukaryotes because they're fungi. But these are cells that contain nuclei and have other complex features such as mitochondria and whatnot. So you see that animals, fungi, plants all exist within this are all eukaryotes. So we'll talk about that a bit more in a bit. But I think it is important to mention that evolutionary biology is essential to understanding any field of biology. So if we go even a little deeper, we can go into organismal biology, which may be what you think about when you think of biology. You think of different, species. So there's an elephant or a mouse or a fruit fly or something like that. So this is essential for biochemistry and also for understanding the power of biochemistry. So there's this guy, Jacques Monod, who said, what is true for E. coli is true for the elephant, which really does hold up when you're thinking in terms of biochemistry. So all the basic processes that occur in E. coli also occur in the elephant, a lot of times in the same exact way. And so this is really important for research as well, because we use a lot of model systems basically. So I have here the biochemistry of every living organism is remarkably similar. And this is to the extent that most of the knowledge about how your human body functions, so how your cells divide, your circadian rhythms, your metabolism, your signaling pathways that allow you to see light and taste and hear things, almost like all that knowledge was first determined in these types of model organisms. So bacteria, such as E. coli, yeast is a really big one for understanding how our cells divide as well. There's also other model organisms like this one called C. elegans, which is just a very small worm, and scientists basically know and have like a name for every single one of its something like 2,000 or 900 cells or something like that in C. elegans. Huge in aging biology, so a lot of aging studies are done in C. elegans. So plant scientists will often just study this one very simple plant called Arabidopsis thaliana, or a rock crest right here. And fruit flies. So fruit flies are really important for understanding our physiology and our biochemistry. So things like developmental biology, cancer biology, and genetics. Those are very active. You can learn a lot just by studying fruit flies, and a lot of it extrapolates exactly 1-to-1 correlation with the human body, and of course mice. So when we're developing vaccines and all sorts of different, determining genetic disorders and stuff like that, you always do that in mice first, or if you're developing new drugs and stuff like that. So organismal biology is very important, and it gives us a chance to show how, and we'll discuss throughout the course, how similar the biochemical processes in something like E. coli holds true for human beings as well. And then we can go even a little deeper into cellular biology. So as I mentioned earlier, the cell is the most simple unit of life. So life doesn't get any simpler than a single cell. And so cells, as I mentioned, also can be delineated into prokaryotes, archaea, and eukaryotes. So here we have a prokaryotic cell right here. So this is a bacteria. And so they are distinguished by certain features. So they, just like eukaryotic cells, they have a cell membrane, which delineates the outside environment from the inside environment. So one thing there's a few things unique about prokaryotes. One is that the DNA, which are these lines here, are just scattered throughout the cell. That's a pretty big difference. This bacteria here also has these little filaments outside of it, and a flagella, which it can use sort of either of those for motility. So this over here is a depiction of eukaryotic cells. So the most obvious difference between eukaryotic cells and prokaryotic cells is this right here, which is the nucleus. So all the genetic information in prokaryotes is housed within the nucleus. But it also has a complex other organelles within the cell. So organelles are just other compartments that are differentiated and have specific functions within the cell. So here we have some what are called mitochondria, which are the powerhouses of the cell. We'll talk a bit more about mitochondria. So this is where a lot of very important metabolism occurs. So all the energy produced in your cell, for the most part, is produced. So all that ATP is produced in the mitochondria. There's also like the Golgi apparatus here in the endoplasmic reticulum. So basically, they make proteins, and then these can get shuttled out and kicked out of the cell from there. So we'll have a whole section on cellular biology in a few weeks. In multicellular eukaryotes, such as ourselves, cells can be very differentiated as well. You can have a lot of different unique cell types with different distinct morphologies and functions. So right here we have neurons. You can see they have these sort of branches here, these axons and these dendrites, and that's how your brain functions. They have these long morphologies, and they communicate with each other, and that allows me to basically be talking to you right now. And here we have a red blood cell, we have a platelet cell, and then we have a white blood cell. And you can see they each have distinct morphologies based on their function. So cellular biology, the most fundamental primary unit of life. And so that brings us finally to biochemistry again. So as I said, biochemistry is the most fundamental of the different biological sciences. So biochemistry basically analyzes the structure and the interaction of different biological macromolecules, or different biological chemicals that are used to sustain life. So we'll talk about that in a second, but biochemistry, there's basically 4 different main classes of biological macromolecules. So there's proteins, lipids, nucleic acids, and carbohydrates. And then there's other small molecules and ions that are also involved in sustaining life. So biochemistry looks at the structure and the interactions of these biological molecules specifically. And that gives you a lot of really, really good information on how the cell works, how the organism works, how the ecosystem works, and so on and so forth. It's really been a very successful discipline, especially throughout the 20th century, which is sort of in its heyday, to explain how biological systems work, because biochemistry is so fundamental. So here we have a protein right here that's binding oxygen, that is myoglobin. So protein, that's what proteins can do. This is ATP, which I mentioned before, which would be a small molecule, but it's a nucleic acid. And so this is the energy currency in your cell. So you eat food, turn it into energy. That means you're turning it into ATP. You burn that energy to, run or, think a thought or anything like that. That's you using burning that ATP energy. So ATP is really fundamental. Lipids is also very important. So this is a lipid vesicle. And so I said that the cell is the most fundamental unit of life. And that's because it can differentiate the outside environment from the inside environment. And that's only attained through the lipid-based membrane around each of your cells. So here is an example of a specific lipid molecule right here. So it's got a hydrophilic head and a hydrophobic tail. And that's basically all you need to form this type of vesicle right here, which allows your cells to form. So right here we have a glucose molecule, which is a carbohydrate. And so this is a really powerful energy molecule. So we'll spend a little time talking about metabolism. So metabolism is a really important part of biochemistry. You can basically trace, okay, I ate a glucose molecule and I want to turn it into energy, into ATP. And through the study of biochemistry, we know every single step from how the energy in that glucose molecule is transformed into a usable form of energy in the form of ATP. And we have DNA right here. So that would be a nucleic acid as well. It's a nucleic acid polymer. So it's a polymer of these types of ATP and similar molecules. And of course, DNA stores the genetic information in your cells that allows you to express a phenotype. So the fact that you're a human and an elephant and is an elephant, and an E. coli and is an E. coli, and a fungus is a fungus is because DNA is because of the differences in the DNA sequence in each of those organisms. I think this is carbohydrates again. So carbohydrates can also be used as storage molecules. So these are starch. Oh yeah, these are starch molecules from a plant. So we talked about how important plant domestication was. It's because we wanted readily available sources of glucose in the form of starch. So this right here are starch granules. And then just here's another protein. So we'll talk a lot about proteins as well. They're very important. And you can tell that this protein over here has a different shape than this protein. That's because proteins do specific jobs in your body, and that's based on the structure of these different proteins. So figuring out the precise structure and how that relates to the function of each and every protein in your body or in any organism is a goal of the study of biochemistry. So biochemistry is the most fundamental of the biological sciences. There's 4 major macromolecules, and we'll talk more about them. And we'll go through each of those molecules now, and we'll look at biochemistry for the this entire course. So as I mentioned, biochemistry is really instrumental to understanding medicine. So modern medicine gleans so much information from just this study of biochemistry. So if you want to understand the chemistry of life, this will provide you with insights into the mechanism of disease. So disease is basically where something in your body has gone wrong. So if you understand the biochemistry of how your body functions normally, you can understand the biochemistry of a disease state and potentially find different ways to manipulate that biochemistry so that you can develop treatments and drugs. So disease mechanisms are illuminated by understanding biochemistry. So right here we have what's called a Lewy body. And so this is a part of your brain called the substantia nigra. And in the disease state, normally there wouldn't be any problems, but in this disease state, you get these little insoluble inclusion bodies called Lewy bodies that are just aggregates of protein, of misfolded proteins, basically. And this can cause the cells to die and for you to develop diseases such as Parkinson's disease. So understanding what a protein is, what a protein misfolding is, and how that can cause the cell to die, and how that cell death leads to Parkinson's disease can help you understand understanding that biochemistry can help you understand the disease. So for instance, this inclusion bodies kills these cells, which produces this molecule here called dopamine, and which I don't know why the E is down here. And then we can understand what is the importance of dopamine in your body. And then we can understand that, well, dopamine comes from this precursor, L-DOPA. And so L-DOPA can then actually be used as a treatment for Parkinson's disease. So this just shows you how understanding biochemistry can help you understand disease. So this protein right here is actually the most abundant protein in your body, and it's called collagen. And so understanding the structure of collagen, you can see that it has these three sort of helices going right here, and those are caused by chemical interactions between these specific chemical groups. Sometimes those chemical groups can be mutated, and then it can cause your bones to be very brittle, a disease called osteogenesis imperfecta. So understanding the structure of collagen and then what happens in disease really gives you some insights into that. And there's also more fundamental. So this is a brain, this is your brain, and this is your brain on of an Alzheimer's disease patient. So biochemistry helps us understand that, and also helps us actually visualize brains in this way, so we can actually understand what's happening in the brain of an Alzheimer's disease patient. And then here we have an insulin receptor bound to the hormone insulin. And so diabetes is a really important disease. And so understanding how diabetes works, and that it's an insulin sensitivity or an inability for your body to make insulin, can help you develop treatments and drugs to help treat diabetes, to help these patients. So biochemistry is super important for understanding all different types of medicine practices, both in human and veterinary medicine. Biochemistry is also really important for nutrition. So I mentioned before that metabolism is a key component of the study of biochemistry. We know when you intake a fat molecule or a glucose molecule or amino acid from a protein from a steak that you ate, we know through years and years of biochemical investigation, we know exactly how those foods are turned into energy and turned into raw materials to help to make more fatty acids and more glucose and more proteins for your own body. So we know that each of these molecules are eventually turned by your body inside the mitochondria. So this means inside the mitochondria, are turned into a different molecule called acetyl-CoA. That is then oxidized, and those electrons are transferred to these molecules. Those are pushed through a collection of proteins called the electron transport chain, and then those electrons are transferred to an oxygen molecule to form water. So this oxygen molecule right here, so this O2 molecule, is basically the point of why you breathe. So every single oxygen molecule, or basically every single oxygen molecule you've ever breathed for your entire life, has been used by this electron transport chain to turn food that you eat into energy. And so so the electron transport chain uses that to pump these protons into this space, and then that sort of actually powers like a dam type thing to turn ADP into that molecule ATP I keep talking about. So ATP is used to burn energy for motion, active transport. That basically means how your neurons work. A lot of your energy is used just for neurons. So like chess players, basically, like high grandmaster chess players, just by sitting and staring at a chessboard and going through all the permutations can actually burn more calories than someone running a marathon, which is pretty interesting because this active transport is so important. Anyway, that's an aside. And also synthesizing other molecules. So that takes energy, and then you can oxidize fuel molecules. So like what I was talking about here, and use that to form new energy. So this is all important to biochemistry, and also things like nutritional deficiency. So through biochemistry, we know that a vitamin C deficiency causes scurvy, and we know exactly why, because it causes a breakdown in that collagen molecule we talked about before. So biochemistry is really important for understanding nutrition as well. And biochemistry is also central to agriculture. So we talked about how agriculture completely changed human society. Well, agriculture, with the onset of biochemistry, agriculture has gotten significantly more sophisticated and allowed us to feed a larger human population than was ever possible at any other time in our collective history. So biochemistry is fundamental to both understanding plant and livestock agriculture. So some examples of this, I mentioned starch composition. So this is one of the first plants ever domesticated. And so, and this basically right here is just a little starch factory. So we understand we can change the composition of that starch if we have an understanding of biochemistry. Same with this corn right here. Oh, so photosynthesis. We have a huge understanding of how photosynthesis works and stuff like that. So we can actually there's goals to try and modify that to increase plant yield. Also, things like livestock nutrition that you can see right here. So knowing what type of nutrition that pigs or goats or chickens or cows need to maximize output is the purview of biochemistry. And we'll also talk about things like insecticides. There's a boll weevil on a cotton plant. Which is so developing insecticides that's been spurred along by biochemistry, and also herbicides. So here's the molecule glyphosate, also known as Roundup. So Roundup could not have been developed and implemented without an understanding of biochemistry, and also disease protection. So this right here is a virus, African swine fever virus. Which is a very complicated virus and is threatening the United States at the moment. But biochemistry is necessary for vaccine and therapeutic development to keep livestock healthy and free of disease. So biochemistry is also integrally important in agriculture. So now we can actually go through the different classes of biomolecules, which are important to biochemistry. And the first class of biomolecules we'll go through are proteins. So proteins are really important in biochemistry and just in sustaining life in general. So what proteins are they're polymers of amino acids that fold into specific three-dimensional structures, and that dictates the function of that specific protein in the cell. So we have these individual molecules called amino acids, and then those are going to be organized and linked covalently into a specific sequence of specific amino acids. So there's 20 different amino acids, and then they'll be in a specific sequence that's specific to that particular protein. And so that specific sequence will fold into a particular three-dimensional structure. And whatever that three-dimensional structure is, the basis of which amino acids are on the outside and the inside and where they're located in three-dimensional space, that's going to dictate the function of that particular protein. And proteins do all sorts of different jobs in your cell. One really important one is catalyzing chemical reactions. So those are called enzymes. So enzymes can take one chemical or two chemicals and combine them or just change the chemical around in different ways. Also, motility is important. So your muscles are made basically entirely of proteins. And if you think of like a bacteria, the flagella on the bacteria that allows it to move around is made out of proteins. Immunity. So antibodies and the different cells involved in antibodies and the recognition of foreign pathogens in your body. That's all involving proteins. Also membrane transport, which is really important to cellular function and neurology and stuff like that. And also structure, just making sure that there's structural integrity to different parts of life. All these are catalyzed by protein. So I already showed you this insulin receptor here. So it has this structure, and the structure right in this region is exactly perfect to bind to insulin to have some sort of effect. This is an antibody right here. So it's going to have a specific three-dimensional structure over in this region here, in this region here, that'll bind to a pathogen and neutralize it. And this is another protein called ATP synthase, which is really large and complicated and made up a bunch of these different polypeptide amino acid sequence chains. And it is basically a molecular machine that'll form ATP in your body. So proteins are really important. We'll come back to them a lot. The second class of biomolecules is nucleic acids. So these do a lot of different things, but the main function of them is to store and convey genetic information. So right here is the DNA double helix. So this stores all of your genetic information. You can replicate it, and it's heritable. It's basically formed from these base pairs, these nucleic acid base pairs. And so you might be familiar with A's, T's, C's, and G's in your DNA sequence, and those are from these different chemical groups that are in the DNA double helix. And the helix is a double helix, so it's able to actually be replicated. So these two strands can come apart, and you can have replication of a different strand on it, and so on and so forth. And that's how you're able to replicate your DNA. Now, nucleic acids, they store genetic information, but they also convey it through a process called the central dogma. And so the central dogma is that you have DNA, which is replicated, and then that DNA is transcribed into a different nucleic acid polymer called RNA. And then that RNA is translated, the code of that RNA is translated into that specific protein amino acid sequence. And so, I had a professor in undergrad who basically told me that the secret of life, or told everyone that the secret of life is getting the right protein in the right concentration to the right place at the right time. And the central dogma, this process here, is basically how that happens. Nucleic acids are also involved in some other processes. So ATP, that energy molecule, that is also a nucleic acid, and they're also involved in cell signaling or communication between cells. So nucleic acids are biomolecule class number 2. The third class of biomolecules are lipids. So lipids, if you're thinking lipids, you could basically think fats, although it's a bit more complicated than that. So these are molecules with a specific characteristic in that they have a hydrophilic or water-loving region or head group. So here is a lipid molecule here. It's got this hydrophilic head group that's going to interact with water, and they have a hydrophobic or water-hating tail. So they're amphipathic, like amphibian. So one side is water-loving, one side is water-fearing. And so a really important thing that allows lipids to do this is to form these bilayer membranes around cells. So here's a pretty cool representation of part of a lipid membrane. So this part right here in gray and red is the lipid bilayer. So this tail region is on the inside, and the head region interacts with these water molecules up here in blue on either side. So this allows the cell to sort of form this lipid bilayer around the cell to, basically to keep the external environment different from the internal environment of the cell. The nucleus in the cell and all these different organelles also are delineated by these lipid bilayers. But lipids are also important for other stuff. They're really important for metabolism. So you eat a lot of fats in your diet. So those can be stored in adipose tissues as these lipid droplets, and you can use those. These lipid molecules are really energy rich because you can basically break off these different carbon groups from this hydrophobic tail and use that to provide energy. So if you're ever, like trapped in your plane goes down in the Andes, or your wagon train gets stuck in the middle of, the wilderness or something, the extent to which you or other people are going to survive is going to be based entirely on how much of these fat deposits that you have. Lipids are also involved in cell signaling. So this molecule down here is actually a steroid molecule that's involved in different like endocrine processes in your body. So these are also considered lipid molecules, even though they don't have they still have a hydrophilic region and a hydrophobic region, but they're a little bit different. So the third class of biomolecules are lipids. And so the last biomolecule class is carbohydrates. So carbohydrates are named that because they're literally carbohydrates. So they have the simplest or the most important one is glucose here, and it has this chemical structure, as you can see right here. And they're made up of like repeating units of CH2O. So you can see how they're carbohydrates. So there's a CH2O. And this atom right here is a carbon, so that's CH2O, CH2O, CH2O. So they all have that sort of type of repeating unit. So these are really energy-rich molecules, and they're used to store and release energy. So if you think of carbohydrates, you probably already think of like bread and pasta and stuff. Those are just they come from starch and stuff, so those are just made up of a bunch of different glucose molecules. We as animals also store glucose in the form of glycogen, which is shown here, which is just these chains of glucose molecules that you store when you don't need a bunch of energy. And then if you go on a run or something, you start breaking down these glucose energy, or if you haven't eaten in a while and your blood glucose level's getting low, then you can break down that glucose. But carbohydrates are used, and so this is a starch molecule right here. But carbohydrates are used for other things as well. So this molecule right here is probably the most abundant biomolecule, or it is the most abundant biomolecule on the planet. It's called cellulose. It's what gives plants basically their structural support. So it's also made out of glucose, just in a little bit different form than glycogen or starch. So if your piece of paper or a newspaper, that's made out of cellulose. And they're also involved in cell signaling. And carbohydrates are also really important in protein modification. So I told you that proteins are polymers made up of amino acids. Well, once a protein is folded in a specific way, it can also be decorated with these carbohydrate units, and that's really important as well. So that's the last biomolecule class. So we had proteins, nucleic acids, lipids, and now carbohydrates. So now we can start getting into the last section of the course, which is where we talk about different, just some examples of what how the class is basically going to be set up, where we talk about different issues that affect society and how biochemistry relates to these different issues. So the obvious first choice is the one that has affected everybody over the past couple of years, and that's COVID-19. So the COVID-19 pandemic, of course, started in December 2019 with an outbreak in China, and really has impacted every single individual and every society on the planet. And so here's some, of course, images of the darkest days of the COVID-19 pandemic. Here is the vaccine that helped us get out of or manage the pandemic. And here's the case number and the deaths overall of the pandemic in general. So I'm not going to go into detail on this right now. You all lived through it, of course, and we'll talk about it in later lectures. But I thought that talking about SARS-CoV-2, the virus, and COVID-19 is a good way to sort of talk about how to reinforce these four different classes of biomolecules. So proteins, nucleic acids, lipids, and carbohydrates. So we'll just do that for a couple minutes. So of course, the COVID-19 pandemic was caused by the virus called SARS coronavirus 2. So this SARS-CoV-2 stands for severe acute respiratory syndrome coronavirus 2, because there was a SARS-CoV-1, which occurred earlier in the 21st century. So here's an electron micrograph of the actual SARS-CoV-2 virus. Here is sort of a diagram of what it looks like, and here's more of a cartoon diagram. And then here is the genome of SARS-CoV-2. So if we just look at the virus, we can already see how these 4 classes of biomolecules are so important and impact your life. So if we look at the SARS-CoV-2 virus, it contains a lipid membrane. So there's a lot of different types of viruses, and SARS-CoV-2 just happens to be a lipid enveloped virus. So it actually has a lipid membrane in this region here. So outside of it are proteins, and then inside is where the genome is held. So this whole region here in blue is actually made out of a lipid bilayer, and that's sort of designated in this picture right here in gray. That membrane has several different proteins embedded in it, the most important of which is the spike protein. So SARS-CoV-2 is a coronavirus, and it's called that because corona is the Latin word for sun. And it's sort of, you can see how it sort of looks like a sun, and that's because it has these spike proteins that stick out from the outside of the protein. And these are what actually recognizes your cell and causes invasion. And it's also what antibodies are going to be recognized by it, and how and it's the protein that's encoded by the mRNA vaccine that you hopefully took at some point. So these right here are the different proteins. There's also some other proteins embedded in that membrane. So you can see here is the S protein, or the spike protein. There's also an envelope protein right here. And there's nucleoproteins that actually surround the genome. And another important point, so I mentioned that carbohydrates often decorate protein. Well, this spike protein here is highly decorated with carbohydrates, and that helps this protein to recognize receptors on the cell that it's trying to invade. And then lastly, of course, this membrane encloses a genome made of the nucleic acid RNA. So SARS-CoV-2 actually doesn't have a DNA genome, it has an RNA genome. And so that's quite interesting. And so the outline of this genome is here. So the genome encodes certain different genes, including that spike protein, as you can see right here. So the genetic information in the spike protein is going to be used to make that protein that's embedded in the lipid membrane. So proteins, nucleic acids, carbohydrates, and lipids are also all really involved in the entry of SARS-CoV-2 into your cells and its replication. So here we have a bit of a too detailed outline of how it enters the cells and how it replicates, but it should be okay for our purposes. So here is the actual virus. It's got the spike and the RNA genome inside of it. So what happens is that the spike protein is going to recognize this receptor on the lipid membrane of your cell. This receptor is going to be made out of protein. So it's just a normal receptor. The virus has just evolved to recognize that specific receptor. That receptor is then going to take the virus inside the cell, and it's going to release that nucleic acid genome into the cell. Now, and it's going to basically hijack the central dogma in your cell. So it's going to turn that RNA into different proteins. And so those proteins are then going to get modified, and it's going to replicate its RNA genome. And then they're going to go through your lipid membrane-bound organelles and form new viruses. So that viruses that have those proteins on the outside and that mRNA genome on the inside. And then they're going to go through a lipid membrane. So that lipid envelope is actually made from lipids in your own cells. And then it's going to be released and probably lyse the cell. So here is a pretty cool illustration of this is the spike protein right here. And this is the receptor that it uses to get into your cell. It's actually called an ACE2 receptor. It functions in our body normally as like a blood pressure modulating receptor. But SARS-CoV-2 will actually interact with it in this specific region right here. So I worked on SARS-CoV-2 through the entire pandemic, and we all paid, or at least I did especially, paid very close attention to when new variants came out in the SARS-CoV-2 genome. So you'll remember there's, alpha and beta, and then there was a delta wave, and now we're in the Omicron wave as of this recording. And so the variants that are the most scary are the ones that interact with this ACE2 receptor. For two reasons. One is because the variants can change the affinity of this protein-protein interaction that allows the virus to get into the cell. Now maybe it can recognize it a bit better and get into the cell a little easier. The other issue is that the antibodies that you get from a vaccine or from a prior infection are going to recognize the part, basically this part right here. And so if there's too many variants or mutations on that specific region, that's going to decrease the affinity between that protein antibody and the protein spike protein. So this is a really good way, SARS-CoV-2, to talk about proteins, nucleic acids, carbohydrates, and lipids. So now we're going to get into the first topic that we're going to talk about when we're talking about biochemistry and society. And so your book goes through this topic specifically. So we're going to talk about the pill. And by the pill, we mean the pill, one of the most important pills that's ever been developed by humans and really changed society in a very significant way. So when someone says the pill, they're usually referring to one of many oral contraceptive medications that prevent pregnancy. So the pill, so a pill that you can take that would basically 100% stop you from getting pregnant. That type of convenience and effective form of birth control was completely unavailable throughout all of human history until the year 1960. So that's a pretty profound thing. So people have been trying not to get pregnant for basically all of human history. And it was not possible until the development of the birth control pill in 1960. So this has had significant impacts on society since. So basically, as long as humans have been getting pregnant, they've probably been developing ways to try not to get pregnant. And we do know a lot about this topic through ancient text and ancient medicines and stuff that developed after the agricultural revolution with the onset of, writing systems like we were talking about before. So a lot of different methods of birth control have been used all throughout history since very early history, with varying degrees of success and safety. So one of the earliest that probably was used before the agricultural revolution is extended breastfeeding. So it is known that when a woman is breastfeeding their child, she basically stops ovulating for the most part. And this can be a pretty effective way to basically space out pregnancies so you do not have too many children all at once that cannot be supported. So the reliability of this method is not ideal. Your book, I think, has some statistics on that, but it's probably something that our early ancestors in pre-agricultural societies had some notion of. After the agricultural revolution, then we have some records of methods that people used to try not to get pregnant, basically. So those include ingestion of different plants and chemicals. So there was in ancient China, women were taking terrible like mercury concoctions, basically, that would stop them from getting pregnant, but could have terrible side effects, including brain damage. There's also coitus interruptus. This is probably another earlier one. So this would be referred to as the pull-out method. So there are some religious texts that sort of forbid this. The Bible, I think, has a story about how this is not how you should not do that. But it can be an effective form of birth control, but obviously it is not foolproof, and probably has been used for a long, long time. The rhythm method is also another method that's been used for a long time. This is basically there are ancient texts where And your book mentions these where people recognized that or hypothesized that women were probably ovulating, although they didn't know the process of that, only a few days a month. So if you avoided sex during those specific times of the cycle, you could decrease the likelihood of getting pregnant. So, but of course, this is not a foolproof method either. There's an old joke. What do you call someone who does the rhythm method? You call them mother. So because it's not a foolproof method at all. And then another popular one were various barrier methods. So basically different substances that can be inserted into the vagina that could have spermicidal properties and keep the woman from getting pregnant. So ancient Egypt has a lot of quite odd ones that are mentioned in those texts, including a mixture of honey, acacia leaves, and lint. And that's basically inserted into the vagina. And acacia leaves are known to have some spermicidal properties, so this might have been a somewhat effective one. A less sort of appetizing one or appealing one would be a mixture of honey, carbonic acid, and crocodile dung. So that was also used in ancient Egypt, but doesn't seem like a very pleasant method for anyone to use. So birth control methods have been around probably for all of human history, and we do have some records from the ancient world. So with the development of the pill can actually kind of be traced back to basically the early to mid 1800s in England. So I remember when I said the Industrial Revolution basically started around this time, around 1790. So there's this guy Malthus who basically was the first person to say, okay, well, we're starting to have this industrial society. The quality of life is starting to go up for most people or for society in a general sense. And then people are able to have more and more children, and their children are surviving later and later. So he predicted that this would cause a population growth that would basically outstrip the resources of a society. So this is Malthus right here. And this is, part of his treatise where he talked about this population control. Now, Malthus opposed birth control as a method because he felt that it was unnatural, but he was actually pretty cruel in how he thought it should be handled. He basically just thought basically just let them starve to death. Don't invest in social programs because we can't afford that many mouths to feed. But don't use birth control. But other people, especially in England at the time, would form what are called Malthusian societies or something like that, or Malthusian foundations, where basically they said, yes, this guy's right. This is a problem, but we should deal with this problem through birth control methods. And so here is sort of a satirical postcard where this woman is saying, "Oh my God, this is a terrible scourge on society. These babies," which they're sort of making fun of this woman. But this would have been a woman who might have belonged to one of these Malthusian societies. So the problem or the need or the desire for effective birth control methods and effective information and scientific research on birth control methods basically in the modern world starts from about this time. Now, unfortunately, this sort of thing was stymied in the United States with the passing of what are called the Comstock Act, which made legal what are called the Comstock laws. So these were laws that outlawed literature being shared through the U.S. Post Office on birth control as obscene. So this guy here, Anthony Comstock, was the U.S. Postal Inspector, and he was a fervent moral opposer of anything obscene. So, like sex toys and pornographic material and stuff like that. But he included laws or information on birth control as obscene information. So this anti-obscenity law was actually passed by Congress in 1873, and it was illegal to distribute information on birth control through the post office. Now, parts of these laws were repealed in the 1920s, but the Comstock laws were not fully repealed That is, you could not freely give information about, they weren't fully repealed, and you couldn't just freely give information about birth control through the U.S. Postal Service until 1972. So this is something that was a reaction at the time, this sort of moral panic that has, effects over many, many years. It took 99 years for it to be fully repealed. Now, one of the main figures in changing societal attitudes about birth control and in developing the pill was this woman, Margaret Sanger, who was born in the later 1800s and lived until I believe the 1950s or 1960s, or definitely in the 1960s, sorry. So this woman, Margaret Sanger, who's depicted here, actually coined the term birth control or popularized it. She also opened the first birth control clinic in the U.S. and formed what became Planned Parenthood. So she was actually arrested under the Comstock laws. So the first clinic she opened was actually closed in 11 days because it was considered that she was spreading obscene information. Now, Margaret Sanger is an interesting and sort of complicated person. She was not a perfect person by any means. She had 2 kids, so she kind of ignored, and a husband she kind of ignored and just kind of left the country and stuff like that. So her detractors sort of attacked her personally on that way. But what Margaret Sanger was very ahead of her time and interested in was that she thought that women needed to be real, full human beings and members of society. They needed effective forms of birth control. So she was one of the first people to say that women should be enjoying sex for sex's sake. Sorry, that was kind of hard to say. And that women should be able to enjoy that, and they're not just vessels for to have as many children as possible. So she fought for years and years and years to help people to get information on birth control. So I have a letter here that I found in a book of a way too young mother, so a 30-year-old woman who already had 11 children and was begging Mrs. Sanger to, so you can take a minute and read and pause the video and read it, but she's basically begging her to keep up her fight and to give her information on birth control because she had medical problems. She had 11 children already, and if she was scared that if she had another kid, she would die and all her kids wouldn't even be taken care of, and just saw nowhere else in society besides this woman, Margaret Sanger, to turn to. So Margaret Sanger, although she was trying to get good information on birth control methods at the time, so condoms and stuff with the invention of vulcanized rubber, and after World War I, condoms did become more available. But one thing she insisted was needed by society was a safe, effective oral contraceptive or oral form of birth control. She would see this as the best way for women to control whether and when they get pregnant. And so Margaret Sanger is a central figure in the rise of birth control advocacy and the rise of the pill. So one of the first steps in actually realizing that dream of finding a safe, effective oral contraceptive was the development of synthetic progesterone, so synthetic hormones. In that time, a lot of stuff like hormones or signaling molecules that are available very low amounts in your body, it was very hard to actually, generate enough of them to make a medicine. Like to get insulin back in the day, if you were diabetic, you'd basically have to get it from a bunch of pig kidneys. But this guy, Carl DeGrassi, was working with Mexican yams and was able to isolate and synthesize these synthetic progesterone drugs. And they could be actually orally bioavailable. So you didn't have to get injected with them. You could just swallow them, and then they would be turned into basically turned into hormones in your body. So one of the first ones that was synthesized is norethistérone. Sorry, I've never said that out loud before. And it is basically, you see its structure right here is a little bit different, but it's very similar to both progesterone and estradiol, which is like estrogen, basically. But it's orally bioavailable. So it acts in your body with like it looks a lot like progesterone and a little bit like estrogen. So what happens with that is when they were giving this to lab animals, basically, they found that progesterone basically inhibits the hypothalamus and the release of certain hormones associated with ovulation and estrogen as well. So if you stimulate high levels of estrogen, it would actually increase the release of these hormones, but they can be inhibited by low levels of estradiol. So these two hormones are right here: FSH, follicle stimulating hormone, and luteinizing hormone. So I'm not going to go into huge details on these, but basically if you take these types of medicine if you take high levels of something that acts like progesterone and low levels of estradiol, it basically keeps you from ovulating. It tricks your body into thinking that you're already pregnant and that you don't need to ovulate. It also messes with the lining of the uterus, which makes sperm less likely to get to an egg, even if you did happen to ovulate. So these synthetic progesterones and this investigation into how they affect ovulation in first lab animals and then humans is really how that's the first steps that were taken towards developing the pill. So these were first developed and synthesized in 1951. And so the first oral contraceptive didn't come on the market until 1960s. And that was through the work of basically, it's been argued, just these four individuals and their associates and teams. So through the efforts of Margaret Sanger, who we already talked about, Gregory Pincus, who's this guy here, John Rock, who is this guy here, and this woman, Catherine McCormick, they developed they developed and put through trials and got to market the first oral contraceptive called Enovid. And that entered the market in 1960. So they basically worked with this synthetic progesterone and estrogens over years and years and years, tested it thoroughly in all sorts of different lab animals, and then worked tirelessly to try and recruit human volunteers who would take the pill and then get it approved by the FDA. And they were ultimately successful in 1960. So Gregory Pincus here was one of the main people who started work on animals to test the effectiveness of using these synthetic steroid molecules. This Catherine McCormick woman inherited a bunch of money from her husband who died and really bankrolled all of this type of research and all of these clinical trials because it was hard to get government money for this sort of thing. And then this guy, John Rock, right here, was instrumental in getting the clinical trials done. So he is a gynecologist, and he was also a Catholic, which is very important, but he believed in the birth control pill. Now, getting something like a birth control pill approved by the FDA was very difficult because you're giving a medication to an otherwise healthy person. It's not like they're on death's door, and then that would make like the approval process less, intensive. But they were able to do enough clinical trials to basically get it to market. But the first thing that it was on the market for in 1960 was it was only approved to regulate the menstrual cycle. But savvy women at the time knew that they could go to the doctor and say, oh, I need to regulate my menstrual cycle, wink, wink, nod, nod. And you could get an effective form of birth control. And of course, the first way that it was, they would just give it to you in a pill bottle like this and say, take a certain amount, and then you stop taking it a certain amount so you can ovulate. And then that was modified using this blister pack, which has been in use ever since. So the first generation of the birth control pill did have some side effects in some people. Sometimes they were kind of rough side effects, but subsequent work over the next couple of years made it very effective and with basically very little side effects as well. So that's Enovid, which is the first oral contraceptive released in 1960. So the availability of the birth control pill in 1960 and after, and the increasing availability of other contraceptive methods that are safe and effective, had an immediate and a profound effect on society. So the women's liberation movement of the 60s and 70s is tied directly to the availability of the pill. There's also a huge increasing participation of women in the workplace, which you can see from this graph right here. So this is women's and men's share of the labor force from 1950 to 2015. So in 1950, the amount of women in the workforce was only around 30%, whereas for men it was 70%. And as you can see, when the birth control pill was developed, that led to a very a quick increase in the amount of women in the workforce, which basically has plateaued, and a decrease of men in the workforce because it lowered the burden on men having to have a job if in a two-person household. Also, the participation of women in higher education. You can see in 1970, only 12% of women were going to college. Actually, I should have found the numbers for before that time, but in 20% of men. And you can see over the subsequent years, the participation of women in with a 4-year college degree has basically exceeded that of men. So this could not be possible without the flexibility that birth control and family planning provides for women. And as I mentioned, the pill was first legally only prescribed for regulating the menstrual cycle. It took a couple of years before doctors were able to prescribe it specifically just as a contraceptive. But birth control actually also had an effect on just women's overall health. So oral contraceptives are also effective at treating things like polycystic ovary syndrome, endometriosis, adenomyosis, and dysmenorrhea, or which is painful periods. Although it's actually the only off-target effect that it's actually been approved for is acne treatment. So birth control also treats acne. So there's a lot of different things that women just regularly had to deal with that did not have a really safe and effective treatment, and birth control provided a way to alleviate these types of syndromes. Although birth control also had some effects that could be perceived as negative, so especially with population issues. So there was really no effective way to control birth or family planning prior to the pill. I mean, there was some, but this is the most effective way. But since the advent of the birth control pill, it's really led to a lower or negative, almost negative growth rate in the United States and elsewhere. So this obviously has positive aspects if you are, on the more Malthusian tip, where you think that, we should perhaps be decreasing the overall global population because of things like climate change and just resources and stuff like that. But modern societies with social safety nets like Social Security are basically designed and set up to require population growth that's going to maintain those social safety nets. So you'll see here is a population diagram of the United States. So you see there's this little bulge around right here from people from the age of about 55 to 70. And that is the baby boomer generation that are currently retiring and entering into old age. And you see after the advent of the birth control pill, there's more of a divot in society. It doesn't look like much here, but these are significant numbers if you take the population as a whole. Now, it's going to cause some issues when there's more people retiring than people able to basically put enough tax dollars into the system to sustain Medicare and Social Security and things like that. So there's a lot of countries that have this overall trend that United States also has, if you don't count in things like migration and immigration, is that you basically are having basically no population growth. So as many people are dying as are being born. In some countries around the world, it's basically negative growth, where you have more people dying or emigrating than are being born or immigrating. So that may be a good thing, but it doesn't bode well for this sort of social safety programs, like I said. So these are complicated issues, and you should just give it some thought as to the pros and cons of things like birth control. So the next topic we're going to talk about is herbicides. So invasive plants, or in other words, weeds, which will compete for resources with your crops. This has been a scourge since basically the beginning of the agricultural revolution. So weeds can significantly decrease yields. And if you're putting so much effort into growing your plants, you don't want some weeds coming around, stealing all the resources from your plants and decreasing your yields. So for almost all of human society, basically weeds had to be dealt with by hand, like these people are doing here in this picture. It wasn't until World War II that we had such an understanding of the biochemistry of plants and the chemistry of different chemicals that there was actually herbicides that were developed that would actually kill weeds without affecting the crops. So basically all plants can be categorized into one of two different categories. So either monocots or dicots. And so there's a few different really important physiological differences between monocots and dicots. A big one, and why it's called a monocot, is when it's for like the seedling. On monocots, there's basically one leaf that emerges, whereas in dicots, there's two leaves that emerges. So generally, all crops that are important to humanity are monocots, so otherwise grasses. A lot of them are in the grass family, whereas all weeds are basically dicots, or broadleaf plants. So here we have monocots over here. You can see that maize, rice, barley, oats, millet, sorghum, wheat, as well as things like orchids and tulips, lilies, also onions, palms, forage grasses like alfalfa for cows and stuff, bamboo, bananas, ginger. These are all monocots, whereas dicots, basically just weeds. So the scientists use this difference between monocots and dicots to actually develop the first herbicides. So biochemists and plant biologists discovered that if you applied a specific plant hormone called an auxin, and a specific auxin called indole-3 acetic acid, or IAA for short. So the chemical structure of IAA is right here. This would cause dicots to die because it basically is telling them to grow faster than they're able to. However, it leaves monocots uninterrupted and totally fine. It does not affect monocots. So this was the first clue that maybe you could develop a chemical that would act like IAA and could be used as an effective herbicide. So to apply these effects of IAA as an herbicide, what scientists wanted to do was develop analogs of IAA. So an analog is like a very similar a chemical that's very similar in structure and would have the same effect, but that would have like a longer half-life and be more stable than IAA. So IAA, if you apply it to a field of plants, it's going to go away very quickly and not be a super effective herbicide. So you want to develop chemicals that act like IAA but are a bit more stable and have a longer half-life. So this was effective around the time of World War II. And some of the first herbicides developed were these two drugs here. So 2,4-dichlorophenoacetic acid, also just called 2,4-D, and 2,4,5-trichlorophenoxyacetic acid, or 2,4,5-T. So this is the chemical structure of 2,4-D. You can see it has these two chlorine groups coming off of this ring. So each of these are carbons. We'll go over that in the next lecture on how to interpret chemical structures. But you can see there's chlorine at the 2 position and the 4 position, whereas in 2,4,5-T, there's chloride at the 2, 4, and 5 position here. Now, if we look at 2,4-D and indole 3 acetic acid, you can see, and you superimpose their structures, you can see they're pretty similar in shape. And so that's why 2,4-D is able to have a similar effect as indole 3 acetic acid. So indole 3 acetic acid, it's a plant hormone. It's going to bind to a specific protein receptor in the plant. So 2,4-D looks close enough to IAA that it's actually also going to bind that same receptor and have basically the same effects. So the development of IAA analogs was essential for the development of these herbicides. Now, herbicides are great if you're trying to increase the yields of crops so you can, make for food, let people have a, more healthy, happy life. But herbicides can also be used for more nefarious purposes, and indeed they were during the Vietnam War. So you've probably heard of this chemical. Your book talks about it. It's called Agent Orange. So this was an herbicide that was made from a 50-50 mixture of those two herbicides we just talked about, 2,4-D and 2,4,5-T. Now, what this was used for in the Vietnam War was to defoliate the jungle to give Americans an advantage. So if you hear a lot of stories about the Vietnam War, there's a lot of soldiers who said, I spent years fighting the enemy. Could have been 10, 20, 30 feet away from them. I never saw the enemy. And that's because the jungle that they were fighting in was so thick. And most of these plants in the jungle are broadleaf plants. So an herbicide made up of a 50-50 mixture of 2,4-D and 2,4,5-T would be effective in removing some of this vegetative cover and then allow soldiers to visualize the enemy and attack them. So Agent Orange was sprayed extensively during the Vietnam War all over the country. And of course, this had a lot of effects because deforestation can have a lot of ecological effects. There's a lot of plants, obviously, and animals that make these jungles their home. And if you deforest it, it's really hard for jungles to come back after deforestation. This is also a problem in the Amazon right now with clear cutting or slash and burn, which is okay on a short scale, but on larger scales, it's the nutrients in the jungle floor are often quite poor. And so when you deforest an entire jungle, it can have horrible ecological effects. So this is an example where something that can be great for society, for agriculture, the development of these highly effective herbicides to increase crop yields, can also be used for nefarious purposes for war and can have horrible ecological effects. Now, if that was the extent of the, consequences of using Agent Orange during the Vietnam War, that would be bad enough. But unfortunately, there was horrible health effects associated with Agent Orange as well. So Agent Orange, when it was synthesized, it's supposed to be just a clean mixture of 2,4-D and 2,4,5-T. So we have 2,4,5-T shown right here. However, when it was synthesized by these chemical companies, it was contaminated with a byproduct of 2,4,5-T called TCDD, or 2,3,7,8 tetrachlorobenzoperodioxin. So this is a dioxin molecule, which you might have heard of before. So dioxin is a known carcinogen, and it can get into the soil, and it's incredibly stable. It does not break down very easily. So it can remain in the water table, in the soil for years and years and years. Now, unfortunately, this did cause a lot of cancer in soldiers who were spraying it on the fields, and it also caused devastating birth defects in children born from people who were exposed. So there's some photos down here of individuals who either were directly affected by the spraying of Agent Orange or who suffered really horrendous birth defects as a result of this dioxin that was present in Agent Orange in Vietnam. So it's a terrible example of something that could have been used only for good purposes, only to increase crop yields. However, just carelessness and thoughtlessness and bad science and bad chemical manufacturing had these horrible effects that has far-reaching impacts on society. And the last topic we're going to talk about in this lecture in biochemistry and society is insecticides. So similarly to herbicides, insecticides are also really important for modern agriculture. So basically any plant that you want to grow for commercial purposes to eat has an insect that wants to eat it and destroy it. So having insecticides that are available to treat plants is really, really useful and really important. And again, throughout most of human history, from the agricultural revolution on, there is very little in the way of being able to control insects. You think of pestilence and locusts in the Bible and in ancient texts and stuff like that. They were so nefarious and so hated by people because you could grow an entire crop, spend the entire season toiling away in the hot sun, growing this thing, and then a swarm of locusts can just come in and eat all of your crops. This still occurs in the world today. Insecticides are also really important for the control of diseases. So there's a lot of diseases that are spread by insect vectors. So one example would be malaria, which is a eukaryotic parasite that is spread by mosquitoes. Another example is typhus, which can be spread by lice on the body lice. So insecticides can also be useful for this. So there's one insecticide that's become quite famous, and it's called DDT. And its structure is shown right here. Again, it's another one with a lot of chloride molecules on it, and it has these benzyl rings, which we'll talk about in the next lecture. So it's called dichloro-diphenyl-trichloroethane, and DDT for short. And this was the first polychlorinated organic insecticide ever synthesized, and it was used heavily during and after World War II for insect control. So here is a picture of a U.S. soldier being sprayed with DDT to control the spread of typhus, but from body lice. And here's an insect spray containing 50% DDT that was used just in the household to destroy insects. So we're not going to go into detail on how DDT kills insects, but basically what DDT does in neurons in insects, not so much in humans, it binds to and messes with voltage-gated ion channels in neurons. So in neurons, you basically have a resting potential usually where there's a negative charge in the inside of the neuron and a positive charge on the outside of the neuron. It achieves this by shuttling positively charged sodium and potassium ions out of the neuron. But when the neuron fires, which basically it gives it an electrical impulse, then those voltage-gated ion channels open up and sodium and potassium ions will flood into the inside of the neuron, giving it what's called an action potential. And so that signal can then be shuttled across the neuron and allow the neuron to communicate with other neurons. So how DDT works in insects is it binds to and opens up these ion channels. So it basically causes the neurons to fire uncontrollably and will eventually cause death in the insect. So if you've ever sprayed an insect with Raid or something like that, like a huge spider, you see that it kind of freaks out and everything. That's because it's messing with its ion channels in its neurons. So it affects insects, but has much less or no effect on humans. Now, DDT was widely used in the United States or around the world, basically, after it was originally put into the market. And this was part of an effort to eradicate malaria in many regions worldwide, and it was actually very effective. So it was also started to be used on a wide range of other agricultural pests and disease transmitting vectors. So there was some good things that obviously came out of the use of DDT. You can see, for instance, here in the southeastern United States, malaria was endemic in these regions until 1946. And part of the reason why it was eradicated in this region was because of the widespread use of DDT. However, if something's successful and people are making a lot of money off of it and you eradicate malaria, you're going to want to use it on all sorts of other insect pests, whether these are agricultural pests or disease-carrying vectors. And that's not always a good thing, because DDT, while it might kill the pest, it might also kill other insects that are either beneficial or just benign. And some of those insects may feed on the insects that you're actually trying to kill. So it could actually cause other problems. But in general, DDT was very useful in eliminating malaria from a lot of different places worldwide. However, we wouldn't be talking about DDT if it didn't have also some negative impacts, and it definitely did. So widespread use of DDT led to significant concentrations in the environment. So there's stories about, plants basically in Los Angeles where they were manufacturing DDT, and it was in huge concentrations and it was basically the excess was being dumped. And it was in huge concentrations just in the marine areas outside of Los Angeles. So what the problem with DDT, and it was first brought to light through this book that this woman, Rachel Carson, wrote, Silent Spring, in 1962. She informed the public about how DDT was affecting birds and other fish. So Silent Spring refers to the fact that one day we may wake up and there's going to be a lot of bird species that are basically missing. So why was DDT affecting birds if they were just spraying it to control insects? Well, it was affecting birds through this process called bioaccumulation. So basically, if DDT gets into the environment, it can affect very small animals like zooplankton, and it can be present in them in very low levels. However, when small fish might eat a lot of zooplankton, and that causes bioaccumulations of DDT and other chemicals in these small fish, then these large fish can eat these smaller fish, which accumulates the DDT even more. And then birds that feed on these larger fish can have really high accumulations of DDT. And this is what people were finding invariably, that these birds of prey, specifically birds that fed on larger fish species, were being highly affected by DDT, and they could actually find them. Basically, there was two things that were really alarming. One was that birds were just dying, so they were having seizures, and just that basically the effect of DDT was that there was enough DDT in these birds' bodies to affect their nervous system, just like the insects. And also it caused thinning of the eggs that these birds laid. So there could be very thin, and then basically you'd have a huge population of like ospreys or bald eagles, but then they would not be able to have enough babies to basically sustain those populations. So those populations were disappearing very quickly. Even more alarming was that they were testing human breast milk and finding huge accumulations of DDT in breast milk. So let's say you're eating trout from Lake Michigan. That was a specific example that actually happened. That then you become the bioaccumulation source for DDT. So DDT, while it was effective in eradicating malaria from the U.S., its widespread use definitely led to environmental contamination with effects specifically on larger fish, birds, and it was being found in humans. And so this ultimately in the 1960s, because of the efforts of Rachel Carson and the book that she wrote, it led to the formation of certain groups, certain citizen groups, specifically this EDF, or Environmental Defense Fund. And the goal of the EDF was specifically to basically sue the government as private citizens on the behalf of the environment. So this is everyone's environment. The environment should have an advocate, and they're saying the DDT is affecting these bird populations. So birds that feed on fish and other carrion, and we're having trouble, like this bald eagle, this peregrine falcon, the California condor, although I don't know if it feeds on fish, but anyway, particularly the bald eagle and the peregrine falcon, their populations were plummeting, and they were basically on the road to extinction based on this widespread use of DDT. So the Environmental Defense Fund was formed from lawyers and scientists who basically were going to court and presenting all this accumulation of scientific evidence that DDT was causing these problems in these bird species. and they were having some success. And then the final success happened with the formation of the Environmental Protection Agency under Nixon in 1970, I believe. And that led through the efforts of the Environmental Protection Agency and the Environmental Defense Fund in a complete ban of DDT in the United States by 1972. So DDT has subsequently been banned in many parts of the world. Right now, it's still only in use in a dozen or so countries. Those are mostly countries with high levels of malaria that they just need some ways to treat them. But there's also problems where insects will become resistant to DDT and stuff like that. So it's a very complicated issue. You should look into it more. I'll leave some videos online on it, but it's a really interesting example where this insecticide was developed, highly effective, using biochemistry, and then biochemists themselves noticed that it was accumulating in the environment, causing all sorts of problems. And then scientists appealed to their government basically to say, hey, we have this information, it needs to be banned. So it's a really good example of the intersection between biochemistry and society. All right, so that concludes our first lecture titled Biochemistry and Society, and we went over a lot of information. But like I said, you don't need to remember any of this for a test. It's just information to help illuminate your day-to-day life and see how biochemistry fits into your everyday life and society at large. So what did we talk about? Well, we went through a whole history of human societies, a very brief history, but a history of human societies. And then we talked about some sociologic terms, just so we're all on the same page. Then we talked about biochemistry and how biochemistry fits in within the disciplines of biology. We also talked about the four major classes of biomolecules, so proteins, nucleic acids, lipids, and carbohydrates. And then we talked a little bit about COVID-19 and the SARS coronavirus to see how just understanding that all biological systems are made up of proteins, nucleic acids, lipids, and carbohydrates really gives you a better understanding of the virus and how it works. And then we moved on to some small examples of how biochemistry and society interact. So we talked a lot about the birth control pill, and then we talked a little bit about insecticides, DDT, and a pesticide, Agent Orange, and how that affected society. So I hope you enjoyed the lecture. So we'll have one of these each week. I understand they're a bit long, but there's only one a week, and you can pause it partway through, start it up again. So that's going to be what we're going to do every week in this course. So make sure that you finish your quiz and your short essay by the end of Sunday on each week. So I look forward to seeing you in the next lecture. Thank you very much. I hope everybody learned a little bit something about biochemistry and society.